This is discovered by Kelvin, Benson and their colleagues using unicellular algae chlorella and seen obliques and radioactive isotope of 14C with a half-life of more than 5,000 years. Cycle is divided into the following three phases: carboxylation, glycolytic reversal and regeneration of RUBP, and are represented in diagram. 13. Photorespiration. Photorespiration is the light-dependent utilization of oxygen and release of carbon dioxide by the photosynthetic organs of a plant. Normally photosynthetic organs do the reverse in the light, i.e., uptake of CO2 and release of O2. Therefore, photorespiration is difficult to demonstrate. It is decrease in the rate of net photosynthesis when oxygen concentration is increased from 2-3% to 21%, sudden increased evolution of O2 when an illuminated green organ is transferred to dark. The site for photorespiration is chloroplast. Peroxisome is required for completing the process. RUBP carboxylase is changed to RUBP oxygenase. This happens at high temperature and high oxygen concentration. Temperature and high oxygen concentration, the affinity of RUBP carboxylase for Ca2 decreases and the affinity for O2 increases. High temperature occurs in tropical areas. 14. C4 dicarboxylic acid pathway, Hatch-Slack pathway, C4 pathway. Hatch-Slack found at a regular mode of CO2 fixation in a number of tropical plants, both monocots and dicots, e.g., maize, sugarcane, sorghum, panicum, pennisetum, atriplex, amaranthus, salsola, etc. Plants are called C4 plants because of the first stable photosynthetic product being a 4-carbon compound. Other plants are C3 plants. C4 plants often live in hot, arid and saline habitats. They have Cran's anatomy. Cran's anatomy, the mesophyll is indifferentiated and its cells occur in concentric layers around vascular bundles having large bundle sheath cells. The mesophyll and bundle sheath cells are connected by plasmodesmata or cytoplasmic bridges. The chloroplasts of the mesophyll cells are smaller. They have well-developed grana and a peripheral reticulum but no starch. The chloroplasts of the bundle sheath cells are large. They have ill-defined grana, a peripheral reticulum and starch grains. In C4 plants, initial fixation of carbon dioxide occurs in mesophyll cells. The primary acceptor of CO2 is phosphoenol pyruvate or PEP. It combines with carbon dioxide in the presence of PEP carboxylase or PEPCO to form oxaloacetic acid or oxaloacetate 4C compound. Oxaloacetic acid is reduced to malic acid or aminated to form aspartic acid. Malic acid or aspartic acid is the bundle sheath translocated to bundle sheath cells through plasmodesmata. Inside the bundle sheath cells they are decarboxylated and deaminated in case of aspartic acid to form pyruvate and CO2. Ca2 is again fixed inside the bundle sheath cells through Calvin cycle. RUBP of Calvin cycle is called secondary or final acceptor of CO2 in C4 plants. Pyruvate is sent back to mesophyll cell. Here, it is changed to phosphoenol pyruvate. Energy is required for this. The same is provided by ATP. The latter is changed into AMP, adenosine monophosphate. Importance: the C4 plants are considered to possess greater photosynthetic efficiency, for they can utilize CO2 until a level of 5 ppm is reached, but the Calvin cycle plants cannot utilize CO2 if the level falls below 40 to 50 ppm. C4 plants can utilize greater light intensities and their temperature optima for photosynthesis exceeds those of C3 plants. The chloroplast plants seem to generate more ATP, which of course makes for improved cellular work. The presence of extensive peripheral reticulum in the chloroplasts of these plants indirectly suggests quicker transport of products and therefore greater utilization of light and CO2. 15. Crassylation acid metabolism CAM. This pathway is found in succulents, mostly members of Crassulaceae, Bryophyllum and Sedum, and a few members of Bromeliaceae, such as pineapple. It is a device designed to meet the pressures of heavy transpiration, arising from their xerophytic environment. These plants obtain their CO2 requirements during the night time when they keep their stomata open and as CO2 buildup occurs, the cell sap turns acidic. This process is called as dark acidification. Following daytime, the stomata remain closed, minimizing transpirational losses, but with the advent of light, the CO2 absorbed during the preceding night is utilized for photosynthetic purposes. The process of light deacidification then occurs. Thus, there is a time lag between absorption and reduction of CO2. This arrangement helps to lessen transpirational stress but is responsible for the extremely slow growth of these plants. 16. Factors influencing photosynthesis. 1. Carbon dioxide. CO2 concentration of the atmosphere is 0.03% or 300 ppm. It is a limiting factor as the available CO2 concentration is lower than the optimum for photosynthesis. Increase in its concentration up to 0.1%, 1000 ppm, increases the rate of photosynthesis in most land plants. A decline is observed beyond it. 2. Intensity. The light intensity at which a plant can achieve maximum amount of photosynthesis is called saturation point. It's -1000 feet candles, 10% of full sunlight in shade plants, 50-70% of full sunlight in C3 sun plants and up to 200% of full sunlight in C4 sun plants, e.g., sugarcane. 3. Quality of light. Maximum photosynthesis occurs in blue, violet and red regions of the light spectrum where most of the absorption is carried out by chlorophylls. Minimum photosynthesis occurs in the green wavelengths. 4. Temperature. It does not influence light reactions of photosynthesis but affects the enzyme-controlled dark reactions. The minimum temperature at which most plants start photosynthesis is 0 degrees minus 5 degrees Celsius, but it can be as low as 20 degrees Celsius for lichens and 35 degrees Celsius for some gymnosperms. The maximum temperature at which photosynthesis can occur is 55 degrees Celsius in some desert plants and 75 degrees Celsius for hot spring algae. The optimum temperature is 10 degrees minus 25 degrees Celsius for C3 plants and 30 degrees 45 degrees Celsius for C4 plants. 5. Oxygen. Small quantity of oxygen is essential for photosynthesis except in some anaerobic bacteria. C3 plants show optimum photosynthesis at low oxygen concentration. The possible reasons are I. Oxygen takes part in oxidation of photosynthetic pigments, intermediates and enzymes in the presence of strong light photooxidation. E. Oxygen is a strong quencher of excited state of chlorophyll. E. Oxygen competes with CO2 for reducing power. However, this effect is not known in C4 plants. IV. It converts RUBP carboxylase to RUBP oxygenase. At very high oxygen content, the rate of photosynthesis begins to decline in all plants. The phenomenon is called Warburg effect. 6. Water. Water supplies H+ and electrons for carbon dioxide fixation. However, less than 1% of the total water absorbed is utilized in photosynthesis. The rest is lost in transpiration. Even a slight increased transpiration reduces the leaf hydration that cuts down photosynthesis by causing stomatal closure and hence decreased CO2 absorption, loss of leaf turgidity, reduced absorption of solar radiations and decreased enzymatic activity. Thus photosynthesis is more sensitive to dehydration than any other metabolic process. 7. Air pollution. Dust and smoke particles present in the atmosphere reduce photosynthesis by reducing light penetration and forming a layer over the plants. Sulfur dioxide, nitrogen oxides, hydrogen fluorides and other air pollutants also decrease photosynthesis. 8. Minerals. Magnesium is a component of chlorophyll. Fe, Cu and Mn are required for synthesis of chlorophyll. Mn and Cl are linked to photolysis of water at PS2. PS phosphate is essential for ATP synthesis. Enzyme activators of photosynthesis include potassium and sulfur. Lower availability of any of these minerals reduces rate of photosynthesis. F1. Enzymes, basic concepts and principles of catalysis. Enzymes, the catalysts of biological systems, are remarkable molecular devices that determine the patterns of chemical transformations. They also mediate the transformation of one form of energy into another. The most striking characteristics of enzymes are their catalytic power and specificity. Catalysis takes place at a particular site on the enzyme called the active site. Nearly all known enzymes are proteins. However, proteins do not have an absolute monopoly on catalysis. The discovery of catalytically active RNA molecules, ribozymes, provides compelling evidence that RNA was an early biocatalyst. The class of macromolecules are highly effective catalysts for an enormous diversity of chemical reactions because of their capacity to specifically bind a very wide range of molecules. By utilizing the full repertoire of intermolecular forces, enzymes bring substrates together in an optimal orientation, the prelude to making and breaking chemical bonds. They catalyze reactions by stabilizing transition states, the highest energy species in reaction pathways. By selectively stabilizing a transition state, an enzyme determines which one of several potential chemical reactions actually takes place. 1. Characteristics of enzymes. Enzymes accelerate reactions by factors of as much as 1,000,000 or more. Indeed, most reactions in biological systems do not take place at perceptible rates in the absence of enzymes. They do not change the equilibrium. The specificity of an enzyme is due to the precise interaction of the substrate with the enzyme. This precision is a result of the intricate three-dimensional structure of the enzyme protein. Like catalyst, the enzymes do not start the chemical reaction, not change its equilibrium, but enhance the rate of reaction. Enzyme lowers the activation energy or energy required to overcome barrier of the reaction. Enzymes are not imported. Every cell produces its own enzymes. They may be active inside the cells, endoenzymes or intracellular enzymes, 70% in mitochondria, or outside the cell, exoenzymes e.g. digestive enzymes. Amylase and protease enzymes are now added to detergent for washing and rennet tablets are used for preparation of cheese. Enzymes have high molecular WEIGHT 6000 kD for bacterial ferredoxin, 250,000 for catalase, 482,000 for urease and 4,600,000 for pyruvate dehydrogenase. Therefore they occur in colloidal state. Opt pH for enzyme action varies for enzyme to enzyme e.g., 2 for pepsin, 6.8 for salivary amylase and 8.5 for trypsin. Most intracellular enzyme works at neutral pH. Enzymes are mobile. High temperature denaturates enzyme. Optimum temperature for enzyme activity is 30 to 400 C for most of animals and 20 to 300 C for plants. Within limits, Q10 equals 2 to 3. The first ribozymes discovered by Sek et al. 1981 in tetrahymena were self-splicing group by intron. It removes introns form newly synthesized RNA. This discovery was RNase-P, ribonuclease-P, Altman et al. 1992, which separates tRNA from hnRNAs at their 5' end. Knoller et al. 1992 discovered, in prokaryotes peptidyltransferase is not a protein but a component of 23S rRNA. Lysozyme is an enzyme that hydrolyzes, breaks down, the cell walls of bacteria. Lysozyme is very common. It is found in saliva, in digestive fluids, even in body secretions. It is one of the first lines of defense against bacterial infection. Lysozyme is a small enzyme, a single polypeptide chain with both alpha helix and beta sheet regions. 6 major classes of enzymes. Class type of reaction example. 1. Oxidoreductases oxidation reduction lactate dehydrogenase. 2. Transferases group transfer nucleoside monophosphate kinase. NMP kinase. 3. Hydrolysis hydrolysis reactions transfer of functional groups to water chymotrypsin. 4. Lyases addition or removal of groups to form double bonds fumarase. 5. Isomerases isomerization intermolecular group transfer triphosphate isomerase 6. Ligases ligation of two substrates at the expense of ATP hydrolysis aminoacyl tRNA synthetase. F3 Many enzymes require cofactors for activity. The catalytic activity of many enzymes depends on the presence of small molecules termed cofactors, although the precise role varies with the cofactor and the enzyme. Such an enzyme without its cofactor is referred to as an apoenzyme. The complete, catalytically active enzyme is. Cofactors can be subdivided into two groups, metals and small organic molecules. The enzyme carbonic anhydrase, for example, requires CN2+ for its activity. Glyphosphorylase, which mobilizes glycogen for energy, requires the small organic molecule pyridoxal phosphate PLP. Cofactors that are small organic molecules are called coenzymes. Often derived from vitamins, coenzymes can be either tightly or loosely bound to the enzyme. If tightly bound, they are called prosthetic groups. Loosely associated coenzymes are more like cosubstrates because they bind to and are released from the enzyme just as substrates and products are. The use of the same coenzyme by a variety of enzymes and their source in vitamins sets coenzymes apart from normal substrates, however. Enzymes that use the same coenzyme are usually mechanistically similar. Enzyme cofactors. Cofactor enzyme. Coenzyme metal. Thiamine hydrogen 2 plus carbonic anhydrase flavin adenine nucleotide monoamine oxidase ZN2 plus carboxypeptidase nicotinamide adenine dinucleotide lactate dehydrogenase Mg2 plus eco RV pyridoxal phosphate glycogen phosphorylase Mg2 plus hexokinase coenzyme 8 CoA acetyl CoA carboxylase Ni2 plus urease biodin pyruvate carboxylase mo nitrate reductase 5. F4. Water-soluble vitamins function as coenzymes. The water-soluble vitamins include ascorbic acid, vitamin C, and a series known as the vitamin B complex. Ascorbate, the ionized form of ascorbic acid, serves as a reducing agent and antioxidant. The vitamin B series comprises components of coenzymes. Note that, in all cases except vitamin C, the vitamin must be modified before it can serve its function. Water-soluble vitamins. Vitamin coenzyme typical reaction type consequences of deficiency. Thiamine B1, thiamine pyrophosphate aldehyde transfer beriberi, weight loss, heart problems, neurological dysfunction. Riboflavin B2, flavin adenine dinucleotide, FAD. Oxidation reduction chiliosis and angular stomatitis. Leaves of the mouth dermatitis pyridoxine B6 pyridoxal phosphate group transfer from amino acid suppression, confusion, convulsions nicotinic acid. Niacin. Nicotinamide adenine dinucleotide, NAD+. Oxidation reduction pellagra dermatitis, depression. Diarrhea pantothenic acid coenzyme A acyl group transfer hypertension biodin lysine complexes. Biocidin. ATP-dependent carboxylation and carboxyl group transfer. Rash about the eyebrows, muscle pain, fatigue, rare. Folic acid tetrahydrofolate transfer of 1 carbon components, thymine synthesis. Anemia, neural tube defects in development. B125. Deoxyadenosyl cobalamin transfer of methyl groups, intermolecular rearrangements. Anemia, pernicious anemia. Methylmalonic acidosis. C-ascorbic acid, antioxidant scurvy, swollen and bleeding gums, subdermal hemorrhages. F5. F5. Enzymes may transform energy from one form into another. In many biochemical reactions, the energy of the reactants is converted with high efficiency into a different form. For example, in photosynthesis, light energy is converted into chemical bond energy through an ion gradient. The free energy contained in small molecules derived from food is converted first into the free energy of an ion gradient and then into a different currency, the free energy of adenosine triphosphate. Enzymes may then use the chemical bond energy of ATP in many ways. The enzyme myosin converts the energy of ATP into the mechanical energy of contracting muscles. Compounds and organelles, which can be thought of as enzymes that move substrates rather than chemically altering them, create chemical and electrical gradients by using the energy of ATP to transport molecules and ions. The molecular mechanisms of these energy transducing enzymes are being unraveled. 1. The free energy change provides information about the spontaneity but not the rate of a reaction. Gibbs free energy, or G, measures amount of energy available to do work, hence, free energy involved in reaction is defined in terms of both heat and entropy. Definition. G equals H T S, where G equals change in free energy, a measure of useful work. H equals change in heat content. S equals change in entropy. T equals absolute temperature in degrees Kelvin. Example, in burning glucose e.g. paper. S equals 13 kcal. G equals. The free energy change of a reaction increment G tells us if the reaction can occur spontaneously. 1. A reaction can occur spontaneously only if increment G is negative. Such reactions are said to be exergonic. 2. A system is equilibrium and no net change can take place if increment G is 0. 3. A reaction cannot occur spontaneously if increment G is positive. An input of free energy is required to drive such a reaction. These reactions are termed endergonic. 2 additional points need to be emphasized. The increment G of a reaction depends only on the free energy of the products, the final state, minus the free energy of the reactants, the initial state. The increment G of a reaction is independent of the path, or molecular mechanism, of the transformation. The mechanism of a reaction has no effect on increment G. For example, the increment G for the oxidation of glucose to CO2 and H2O is the same whether it occurs by combustion in vitro or by a series of enzyme-catalyzed steps in a cell. The increment G provides no information about the rate of a reaction. A negative increment G indicates that a reaction can occur spontaneously, but it does not signify whether it will proceed at a perceptible rate. As will be discussed shortly, the rate of a reaction depends on the free energy of activation, increment G, which is largely unrelated to the increment G of the reaction. 2. Enzymes alter only the reaction rate and not the reaction equilibrium. Because enzymes are such superb catalysts, it is tempting to ascribe to them powers that they do not have. An enzyme cannot alter the laws of thermodynamics and consequently cannot alter the equilibrium of a chemical reaction. This inability means that an enzyme accelerates the forward and reverse reactions by precisely the same factor. Consider the interconversion of A and B. Suppose that, in the absence of enzyme, the forward rate constant Kf is 10 to 4 S1 and the reverse rate constant Kr is 10 to 6 S1. The equilibrium constant K is given by the ratio of these rate constants. The equilibrium concentration of B is 100 times that of A, whether or not enzyme is present. However, it might take considerable time to approach this equilibrium without enzyme, whereas equilibrium would be attained rapidly in the presence of a suitable enzyme. Enzymes accelerate the attainment of equilibria but do not shift their positions. The equilibrium position is a function only of the free energy difference between reactants and products. 3. Enzymes accelerate reactions by facilitating the formation of the transition state. The energy difference between reactants and products accounts for the equilibrium of the reaction, but enzymes accelerate how quickly this equilibrium is attained. A chemical reaction of substrate S to form product P goes through a transition state S that has a higher free energy than does either S or P. The double dagger denotes a thermodynamic property of the transition state. The transition state is the most seldom occupied species along the reaction pathway because it is the one with the highest free energy. The difference in free energy between the transition state and the substrate is called the Gibbs free energy of activation or simply the activation energy, symbolized by increment G. Enzymes accelerate reactions by decreasing increment G. The activation energy. The combination of substrate and enzyme creates a new reaction pathway whose transition state energy is lower than that of the reaction in the absence of enzyme. Lower activation energy means that more molecules have the required energy to reach the transition state. Increasing the activation barrier is analogous to lowering the height of a high jump bar. More athletes will be able to clear the bar. The essence of catalysis is specific binding of the transition state. 4. The formation of an enzyme-substrate complex is the first step in enzymatic catalysis. The catalytic power of enzymes comes from their bringing substrates together in favorable orientations to promote the formation of the transition states in enzyme-substrate S complexes. The substrates are bound to a specific region of the enzyme called the active site. Most enzymes are highly selective in the substrates that they bind. Indeed, the catalytic specificity of enzymes depends in part on the specificity of binding. F6. The active sites of enzymes. The active site of an enzyme is the region that binds the substrates and the cofactor, if any. It also contains the residues that directly participate in the making and breaking of bond. These residues are called the catalytic groups. In essence, the interaction of the enzyme and substrate at the active site promotes the formation of the transition state. The active site is the region of the enzyme that most directly lowers the increment G. The reaction, which results in the rate enhancement characteristic of enzyme action. Although enzymes differ widely in structure, specificity, and mode of catalysis, a number of generalizations concerning their active sites can be stated. 1. The active site cleft formed by groups that come from different parts of the amino acid sequence. Indeed, residues far apart in the sequence may interact more strongly than adjacent residues in the amino acid sequence. In lysozyme that degrades the cell walls of some bacteria, the important groups in the active site are contributed by residues numbered 35, 52, 62, 63, 101, and 108 in the sequence of the 129 amino acids. 2. The active site takes up a relatively small part of the total volume of an enzyme. Most amino acid residues in an enzyme are not in contact with the substrate, which raises the intriguing question of why enzymes are so big. Nearly all enzymes are made up of more than 100 amino acid residues, which gives them a mass greater than 10 kd and a diameter of more than 25 A. The extra amino acids serve as a scaffold to create the three-dimensional active site from amino acids that are far apart in the primary structure. Amino acids near to one another in the primary structure are often sterically constrained from adopting the structural relations necessary to form the active site. In proteins, the remaining amino acids also constitute regulatory sites, sites of interaction with other proteins, or channels to bring the substrates to the active sites. 3. Active sites are clefts or crevices. In all enzymes of known structure, substrate molecules are bound to a cleft or crevice. Water is usually excluded unless it is a reactant. The polar character of much of the cleft Water is usually excluded unless it is a reactant. The nonpolar character of much of the cleft enhances the binding of substrate as well as catalysis. Nevertheless, the cleft may also contain polar residues. In the nonpolar microenvironment of the active site, certain of these polar residues acquire special properties essential for substrate binding or catalysis. The internal positions of these polar residues are biologically crucial exceptions to the general rule that polar residues are exposed to water. 4. Substrates are bound to enzymes by multiple weak attractions. S complexes usually have helium constants that range from 10 to 2 to 10 to 8 m, corresponding to free energies of interaction ranging from about -3 to -12 kilocalories mole -1, from -13 to -50 kilojoules mole -1. The non-covalent interactions in S complexes are much weaker than covalent bonds, which have energies between -50 and -110 kilocalories mole -1, between -210 and -460 kilojoules mole -1. Electrostatic interactions, hydrogen bonds, van der Waals forces, and hydrophobic interactions mediate reversible interactions of biomolecules. Van der Waals forces become significant in binding only when numerous substrate atoms simultaneously come close to many enzyme atoms. Hence, the enzyme and substrate should have complementary shapes. The original character of hydrogen bonds between enzyme and substrate often enforces a high degree of specificity, as seen in the RNA-degrading enzyme ribonuclease. 5. The specificity of binding depends on the precisely defined arrangement of atoms in an active site. These enzyme and the substrate interact by means of short-range forces that require close contact. The substrate must have a matching shape to fit into the site. Neil Fisher's analogy of the lock and key, expressed in 1890, has proved to be highly stimulating and fruitful. Now know that enzymes are flexible and that the shapes of the active sites can be markedly modified by the binding of substrate, as was postulated by Daniel E. Koshland Jr. in 1958. The active sites of some enzymes assume a shape that is complementary to that of the transition state only after the substrate is bound. This process of dynamic recognition is called induced fit. F7. The Michaelis-Menten model accounts for the kinetic properties of many enzymes. The primary function of enzymes is to enhance rates of The Michaelis-Menten model accounts for the kinetic properties of many enzymes. The primary function of enzymes is to enhance rates of reaction so that they are compatible with the needs of the organism. To understand how enzymes function, we need a kinetic description of their activity. For many enzymes, the rate of catalysis VO, which is defined as the number of moles of product formed per second, varies with the substrate concentration S. The rate of catalysis rises linearly as substrate concentration increases and then begins to level off and approach a maximum at higher substrate concentrations. Before we can accurately interpret this graph, we need to understand how it is generated. Consider an enzyme that catalyzes the S to P by the following pathway. Michaelis-Menten equation. The meaning of Km is evident from equation 23. When S equals Km, then V0 equals V max 2. Thus, Km is equal to the substrate concentration at which the reaction rate is half its maximal value. Km is an important characteristic of an enzyme-catalyzed reaction and is significant for its biological function. 1. The significance of Km and Vmax values. The Michaelis constant, Km, and the maximal rate, Vmax, can be readily derived from rates of catalysis measured at a variety of substrate concentrations if an enzyme operates according to the simple scheme given in equation 23. The area of Km and Vmax is most commonly achieved with the use of curve-fitting programs on a computer. See the appendix to this chapter for alternative means of determining Km and Vmax. The Km values of enzymes range widely. For most enzymes, Km lies between 10 to 1 and 10 to 7 m. The Km value for an enzyme depends on the particular substrate and on environmental conditions such as pH, temperature, and ionic strength. The Michaelis constant, Km, has two meanings. First, Km is the concentration of substrate at which half the active sites are filled. Thus, Km provides a measure of the substrate concentration required for significant catalysis to occur. In fact, many enzymes, experimental evidence suggests that Km provides an approximation of substrate concentration in vivo. When the Km is known, the fraction of sites filled, Fs, at any substrate concentration can be calculated from. Second, Km is related to the rate constants of the individual steps in the catalytic scheme. Km is defined as K-1 plus K2, K1. Consider a limiting case in which K-1 is much greater than K2. Under such circumstances, the S complex dissociates to E and S much more rapidly than product is formed. Under these conditions, K-1 greater than K2. The dissociation constant of the S complex is given by. In other words, Km is equal to the dissociation constant of the S complex if K2 is much smaller than K-1. Condition is met. Km is a measure of the strength of the S complex. A high Km indicates weak binding, a low Km indicates strong binding. It must be stressed that Km indicates the affinity of the S complex only when K-1 is much greater than K2. Maximal Vmax reveals the turnover number of an enzyme, which is the number of substrate molecules converted into product by an enzyme molecule in a unit time when the enzyme is fully saturated with substrate. It is equal to the kinetic constant K2, which is also called Kcat. Rate, Vmax, reveals the turnover number of an enzyme if the concentration of active sites, ET, is known. For example, a 10 to 6 m solution of carbonic anhydrase catalyzes the formation of 0.6 m H2 per second when it is fully saturated with substrate. Hence, K2 is 6 times 10 5s-1. This turnover number is one of the largest known. Each catalysis reaction takes place in a time equal to 1 K2, which is 1.7 microseconds for carbonic anhydrase. The numbers of most enzymes with their physiological substrates fall in the range from 1 to 10 per second. 2. Vmax and Km can be determined by double reciprocal plots. Ability of computers, the determination of Km and Vmax values required algebraic manipulation of the basic Michaelis-Menten equation. Because Vmax is approached asymptotically, it is impossible to obtain a definitive value from a typical Michaelis-Menten plot. Because Km is the concentration of substrate at Vmax 2, it is likewise impossible to determine an accurate value of Km. However, Vmax can be accurately determined if the Michaelis-Menten equation is transformed into one that gives a straight line plot. Taking the reciprocal of both sides of equation 23 gives a plot of 1 V0 is 1 S called a Lineweaver-Burk double reciprocal plot, yields a straight line with an intercept of 1 Vmax and a slope of Km Vmax. Figure, the intercept on the x-axis is -1 Km. Double reciprocal plots are especially useful for distinguishing between competitive and non-competitive inhibitors. In competitive inhibition, the intercept on the axes of the plot of 1 V0 versus 1 S is the same in the presence and in the absence of inhibitor, although the slope is increased. Figure 1. Intercept is unchanged is because a competitive inhibitor does not alter Vmax. Sufficiently high concentration, virtually all the active sites are filled by substrate, and the enzyme is fully operative. The increase in the slope of the 1 V0 versus 1 S plot indicates the strength of binding of competitive inhibitor. In the presence of a competitive inhibitor, equation 31 is replaced by int I is the concentration of inhibitor and Ki is the dissociation constant of the enzyme inhibitor complex. E plus I equals EI Ki equals EIA. In other words, the slope of the plot is increased by the factor 1 plus I Ki in the presence of a competitive inhibitor. In non-competitive inhibition, the inhibitor can combine with either the enzyme or the enzyme-substrate complex. In pure non-competitive inhibition, the values of the dissociation constants of the inhibitor and enzyme and of the inhibitor and enzyme-substrate complex are equal. The value of Vmax is decreased to a new value called Vapmax, and so the intercept on the vertical axis is increased. The new slope, which is equal to Km Vapmax, is larger by the same factor. In contrast with Vmax, Km is not affected by pure non-competitive inhibition. The maximal velocity in the presence of a pure non-competitive inhibitor, Vmax, is given by F8. Allosteric enzymes. The Michaelis-Menten model has greatly assisted the development of enzyme chemistry. Its virtues are simplicity and broad applicability. However, the Michaelis-Menten model cannot account for the kinetic properties of many enzymes. An important group of enzymes that do not obey Michaelis-Menten kinetics comprises the allosteric enzymes. These enzymes consist of multiple subunits and multiple active sites. The allosteric enzymes have an allosteric site in addition to the active site. Allosteric is derived from the Greek word allo and steric, allo equals other, steric equals site. An effector molecule combined with this site and can either activate or deactivate the enzyme molecule. The effector molecule is termed as modulator, regulator or ligand. The effector molecule may be either activators or inhibitors. The activators are generally one of the substrate or cofactors. In most cases the allosteric inhibitors are the end products of the metabolic pathways, in which particular enzyme is participating. Thus, this kind of enzyme inhibition is also called as feedback inhibition or end product inhibition or retro inhibition. The effect of allosteric is reversible. When they are withdrawn the enzyme resumes its original activity. Allosteric enzymes often display sigmoidal plots of the reaction velocity V0 versus substrate concentration S rather than the hyperbolic plots predicted by the Michaelis-Menten equation. Two models explain the kinetic behavior of allosteric enzymes. The sigmoidal dependence of V0 on S reflects subunit cooperativity, and has inspired two models to explain these cooperative interactions. Its model, the symmetry model proposed by Jacques Monod and colleagues in 1965, an allosteric enzyme can exist in only two conformations, active and inactive. Figure A. All subunits are in the active form or all are inactive. Every substrate molecule that binds increases the probability of a transition from the inactive to the active state. In the sequential model, figure B, proposed by Koshland in 1966, there are still two conformations, but subunits can undergo the conformational change individually. Binding of substrate increases the probability of the conformational change. A conformational change in one subunit makes a similar change in an adjacent subunit, as well as the binding of a second substrate molecule, more likely. There are more potential intermediate states in this model than in the symmetry model. The two models are not mutually exclusive. The symmetry model may be viewed as a all or none, limiting case of a sequential model. The precise mechanism of allosteric interaction has not been established. Different allosteric enzymes may have different mechanisms for cooperative interactions. In allosteric enzymes, the binding of substrate to one active site can affect the properties of other active sites in the same enzyme molecule. The outcome of this interaction between subunits is that the binding of substrate becomes cooperative. That is, the binding of substrate to one active site of the enzyme facilitates substrate binding to the other active sites. Such cooperativity results in a sigmoidal plot of V0 versus S. In addition, the activity of an allosteric enzyme may be altered by regulatory molecules that are reversibly bound to specific sites other than the catalytic sites. The catalytic properties of allosteric enzymes can thus be adjusted to meet the immediate needs of a cell. For this reason, allosteric enzymes are key regulators of metabolic pathways in the cell. For e.g., in threonine metabolism pathway the end product isoleucine acts as inhibitor and binds to enzyme of first step, threonine deaminase. Aspartate transcarbamylase ATPase, one of the best understood allosteric enzymes, catalyzes the synthesis of N-carbamyl-aspartate, the first intermediate in the synthesis of pyrimidines. ATPase is feedback inhibited by cytidine triphosphate, CTP, the final product of the pathway. ATP reverses this inhibition. ATPase consists of separable catalytic C3 subunits which bind the substrates and regulatory R2 subunits which bind CTP and ATP. The inhibitor effect of CTP, the stimulatory action of ATP, and the cooperative binding of substrates are mediated by large changes in quaternary structure. On binding substrates, the C3 subunits of the C6R6 enzyme move apart and reorient themselves. Positive cooperativity. Positive cooperativity means that the enzyme is much more sensitive to changes in the amount of substrate available to it. Consider a situation such as glucose breakdown. You'll realize that the amount of glucose being degraded for energy will vary greatly with the physiological situation of the cell. If it needs a great deal of energy, it will be degrading a lot of glucose. If not, it will be degrading very little or none at all. The enzyme pathway will therefore have to cope with big differences in the amount of substrate that they have to convert and the greater sensitivity to substrate concentration produced by positive substrate. On binding substrates, the C3 subunits of the ATPase consists of separable catalytic C3 subunits which bind the substrates and regulatory R2 subunits which bind CTP and ATP. The ATP, the stimulatory action of ATP, and the cooperative binding of substrates are mediated by large changes in quaternary structure. On binding substrates, the C3 subunits of the C6R6 enzyme move apart and reorient themselves. Positive cooperativity. Positive cooperativity means that the enzyme is much more sensitive to changes in the amount of substrate available to it. If you consider a situation such as glucose breakdown, you'll realize that the amount of glucose being degraded for energy will vary greatly with the physiological situation of the cell. If it needs a great deal of energy, it will be degrading a lot of glucose. If not, it will be degrading very little or none at all. In the breakdown pathway will therefore have to cope with big differences in the amount of substrate that they have to convert, and the greater sensitivity to substrate concentration produced by positive substrate cooperativity will be of great assistance to this. Thus, in positive cooperativity, binding of substrate to any one active site increase the chance of binding substrate to other sites. Negative cooperativity. This means that an enzyme is much less sensitive to substrate change and almost becomes independent of the concentration of substrate. Useful with a substrate, such as a coenzyme perhaps, whose concentration may change due to reactions in the cell which are not directly related to the pathway in which our enzyme is involved. May well be an advantage in these circumstances if the enzyme does not react to changes in that substrate. Here, binding of substrate to any one subunit decreases the affinity of enzyme for other substrates. F9. Enzymes. Enzymes can be inhibited by specific molecules, inhibitors. The activity of many enzymes can be inhibited by the binding of specific small molecules and ions. This means of inhibiting enzyme activity serves as a major control mechanism in biological systems. The regulation of allosteric enzymes typifies this type of control. In addition, many drugs and toxic agents act by inhibiting enzymes. Inhibition by particular chemicals can be a source of insight into the mechanism of enzyme action. Specific inhibitors can often be used to identify residues critical for catalysis. The value of transition state analogs as potent inhibitors will be discussed shortly. Enzyme inhibition can be either reversible or irreversible. An irreversible inhibitor dissociates very slowly from its target enzyme because it has become tightly bound to the enzyme, either covalently or non-covalently. Some irreversible inhibitors are important drugs. It acts by covalently modifying the enzyme transpeptidase, thereby preventing the synthesis of bacterial cell walls and thus killing the bacteria. Aspirin acts by covalently modifying the enzyme cyclooxygenase, reducing the synthesis of inflammatory signals. Reversible inhibition, in contrast with irreversible inhibition, is characterized by a rapid dissociation of the enzyme inhibitor complex. Competitive inhibition, an enzyme can bind substrate forming an S complex or inhibitor A, but not both ESI. The competitive inhibitor resembles the substrate and binds to the active site of the enzyme. The substrate is thereby prevented from binding to the same active site. A competitive inhibitor diminishes the rate of catalysis by reducing the proportion of enzyme molecules bound to a substrate. At any given inhibitor concentration, competitive inhibition can be relieved by increasing the substrate concentration. Under these conditions, the substrate outcompetes the inhibitor for the active site. Methotrexate is a structural analog of tetrahydrofolate, a coenzyme for the enzyme dihydrofolate reductase, which plays a role in the biosynthesis of purines and pyrimidines. It binds dihydrofolate reductase 1000-fold more tightly than the natural substrate and inhibits nucleotide-based synthesis. It is used to treat cancer. Many antibacterial drugs work on principle of CPM peptide inhibition for e.g. Sulfa drugs are believed to be competitive inhibitor of enzyme metabolizing p-aminobenzoic acid PABA. PABA is essential vitamin acting as coenzyme for many bacterial enzymatic reaction and structurally similar to sulfanilamide, a sulfa drug. Similarly, malonate is structurally similar to succinate, the substrate for pyruvate dehydrogenase, and thus competes for enzyme. Inhibition, which also is reversible, the inhibitor and substrate can bind simultaneously to an enzyme molecule at different binding sites. A non-competitive inhibitor acts by decreasing the turnover number rather than by diminishing the proportion of enzyme molecules that are bound to substrate. Non-competitive inhibition, in contrast with competitive inhibition, cannot be overcome by increasing the substrate concentration. Such type of inhibition is shown by allosteric enzymes which are regulated by feedback mechanism. A complex pattern, called mixed inhibition, is produced when a single inhibitor both hinders the binding of substrate and decreases the turnover number of the enzyme. 1. Competitive and non-competitive inhibition are kinetically distinguishable. Can we determine whether a reversible inhibitor acts by competitive or non-competitive inhibition? Let us consider only enzymes that exhibit Michaelis-Menten kinetics. Elements of the rates of catalysis at different concentrations of substrate and inhibitor serve to distinguish the two types of inhibition. In competitive inhibition, the inhibitor competes with the substrate for the active site. The dissociation constant for the inhibitor is given by increasing the amount of substrate can overcome the inhibition. Vmax can be attained in the presence of a competitive inhibitor. The hallmark of competitive inhibition is that it can be overcome by a sufficiently high concentration of substrate. However, the apparent value of Km is altered. The effect of a competitive inhibitor is to increase the apparent value of Km. This new value of Km, called Kapm, is numerically equal to where I is the concentration of inhibitor and Ki is the dissociation constant for the enzyme inhibitor complex. As the value of I increases, the value of Kapm increases. In the presence of a competitive inhibitor, an enzyme will have the same Vmax as in the absence of an inhibitor. In non-competitive inhibition, substrate can still bind to the enzyme inhibitor complex. However, the enzyme inhibitor substrate complex does not proceed to form product. The value of Vmax is decreased to a new value called Vapmax while the value of Km is unchanged. Why is Vmax lowered while Km remains unchanged? In essence, the inhibitor simply lowers the concentration of functional enzyme. The remaining enzyme behaves like a more dilute solution of enzyme. Vmax is lower, but Km is the same. Non-competitive inhibition cannot be overcome by increasing the substrate concentration. F10. Catalytic strategies. A few basic catalytic principles are used by many enzymes. Enzymatic catalysis begins with substrate. Enzymatic catalysis begins with substrate binding. Binding energy is the binding energy is the free energy released in the formation of a large number of weak interactions between the enzyme and the substrate. We can envision this binding energy as serving two purposes. It establishes substrate specificity and increases catalytic efficiency. Only substrate can participate in most or all of the interactions with the enzyme and thus maximize binding energy, accounting for the exquisite substrate specificity exhibited by many enzymes. Furthermore, the full complement of such interactions is formed only when the substrate is in the transition state. Interactions between the enzyme and the substrate not only favor substrate binding but stabilize the transition state, thereby lowering the activation energy. The binding energy can also promote structural changes in both the enzyme and the substrate that facilitate catalysis, a process referred to as induced fit. Enzymes commonly employ one or more of the following strategies to catalyze specific reactions. 1. Covalent catalysis. In covalent catalysis, the active site contains a reactive group, usually a powerful nucleophile that becomes temporarily covalently modified in the course of catalysis. The proteolytic enzyme chymotrypsin provides an excellent example of this mechanism. 2. General acid-base catalysis. In general acid-base catalysis, a molecule other than water plays the role of a proton donor or acceptor. Chymotrypsin uses a histidine residue as a base catalyst to enhance the nucleophilic power of serine. 3. Metal ion catalysis. Metal ions can function catalytically in several ways. A metal ion may serve as an electrophilic catalyst, stabilizing a negative charge on a reaction intermediate. Alternatively, the metal ion may generate a nucleophile by increasing the acidity of a nearby molecule, such as water in the hydration of CO2 by carbonic anhydrase. Finally, the metal ion may bind to substrate, increasing the number of interactions with the enzyme and thus the binding energy. This strategy is used by NMP kinases. 4. Catalysis by approximation. Many reactions include two distinct substrates. In such cases, the reaction rate may be considerably enhanced by bringing the two substrates together along a single binding surface on an enzyme. NMP kinases bring two nucleotides together to facilitate the transfer of a phosphoryl group from one nucleotide to the other. F1. Regulation of enzymatic activity. The activity of proteins, including enzymes, often must be regulated so that they function at the proper time and place. The biological activity of proteins is regulated in four principal ways. 1. Allosteric control. Allosteric proteins contain distinct regulatory sites and multiple functional sites. Regulation by small signal molecules is a significant means of controlling the activity of many proteins. The binding of these regulatory molecules at sites distinct from the active site triggers conformational changes that are transmitted to the active site. Allosteric proteins show the property of cooperativity. Activity at one functional site affects the activity at others. As a consequence, a slight change in substrate concentration can produce substantial changes in activity. Proteins displaying allosteric control are thus information transducers. Their activity can be modified in response to signal molecules or to information shared among active sites. The two best understood allosteric proteins are the enzyme aspartate transcarbamylase ATCase and the oxygen-carrying protein hemoglobin. Aspartate transcarbamylase of the first step in pyrimidine biosynthesis is inhibited by cytidine triphosphate, the final product of that biosynthesis, in an example of feedback inhibition. Binding of O2 by hemoglobin is cooperative and is regulated by H+, CO2 and 2,3-bisphosphoglycerate 2,3-BPG. Two multiple forms of enzymes. ISA, or isoenzymes, provide an avenue for varying regulation of the same reaction at distinct locations or times. Isoenzymes are homologous enzymes within a single organism that catalyze the same reaction but differ slightly in structure and more obviously in Km and Vmax values, as well as regulatory properties. Often, isozymes are expressed in a distinct tissue or organelle or at a distinct stage of development. 3. Reversible covalent modification. The catalytic properties of many enzymes are markedly altered by the covalent attachment of a modifying group, most commonly a phosphoryl group. ATP serves as the phosphoryl donor in these reactions, which are catalyzed by protein kinases. The removal of phosphoryl groups by hydrolysis is catalyzed by protein phosphatases. Cyclic AMP serves as an intracellular messenger in the transduction of many hormonal and sensory stimuli. Cyclic A switches on protein kinase A (pKa), a major multifunctional kinase, by binding to the regulatory subunit of the enzyme, thereby releasing the active catalytic subunits of pKa. In labs of CAMP, the catalytic sites of pKa are occupied by pseudosubstrate sequences of the regulatory subunit. 4. Proteolytic activation. The enzymes controlled by some of these mechanisms cycle between active and inactive states. A different regulatory motif is used to irreversibly convert an inactive enzyme into an active one. Many enzymes are activated by the hydrolysis of a few or even one peptide bond in inactive precursors called zymogens or proenzymes. This regulatory mechanism generates digestive enzymes such as chymotrypsin, trypsin, and pepsin. Caspase, which are proteolytic enzymes that are the executioners in programmed cell death, or apoptosis, are proteolytically activated from the procaspase form. Blood clotting is due to a remarkable cascade of zymogen activations. Digestive and clotting enzymes are switched off by the irreversible binding of specific inhibitory proteins that are irresistible lures to their molecular prey. F12. Isoenzymes, abenzymes and riboenzymes. 1. Isozymes or isoenzymes. Enzymes that differ in amino acid sequence yet catalyze the same reaction. Usually, these enzymes display different kinetic parameters, such as Km, or different regulatory properties. They are encoded by different genetic loci, which usually arise through gene duplication and divergence. Isozymes differ from allozymes, which are enzymes that arise from allelic variation at one gene locus. Isozymes often be distinguished from one another by biochemical properties such as electrophoretic mobility as they differ from each other in electrical charges. The existence of isozymes permits the fine-tuning of metabolism to meet the particular needs of a given tissue or developmental stage. Consider the example of lactate dehydrogenase, LDH, an enzyme that functions in anaerobic glucose metabolism and glucose synthesis. Can be have two isozymic polypeptide chains for this enzyme, the H isozyme highly expressed in heart and the M isozyme found in skeletal muscle. The amino acid sequences are 75% identical. The functional enzyme is tetrameric, and many different combinations of the two subunits are possible. The H4 isozyme, found in the heart, has a higher affinity for substrates than does the M4 isozyme. The two isozymes also differ in that high levels of pyruvate allosterically inhibit the H4 but not the M4 isozyme. The other combinations, such as H3M, have intermediate properties depending on the ratio of the two kinds of chains. Isoenzymes and separation of taxa, that is, chemotaxonomic studies and catalysis of reversible reaction. One form of enzyme can catalyze reaction in one direction, while other form of the same enzyme can catalyze the reaction in opposite direction. 2. Abzyme. In 1969, William Jenks proposed that antibodies, specific for the transition state of a chemical reaction, should have catalytic power. This incisive prediction was realized in 1986 when the laboratories of Richard Lerner and Peter Schultz found that the catalytic antibodies could be produced by using transition state analogues as immunogens. Preparation of an antibody that catalyzed the insertion of a metal ion into a porphyrin nicely illustrates this experimental approach. Ferrochelatase, the final enzyme of biosynthetic pathway for the production of heme, catalyzed the insertion of Fe++ into porphyrin LX. The nearly planar porphyrin must be bent for iron to enter. N-methylprotoporphyrin is a potent inhibitor of ferrochelatase. This compound resembles the transition state because N-alkylation forces the porphyrin to bend. Moreover, it was known that N-alkylporphyrin chelate metal ion 104 times than their unalkylated counterparts. Bending increases the exposure of lone pairs of electron of the pyrrole nitrogen to solvent, which facilitates chelation. Indeed, an effective catalyst was produced using N-alkylporphyrin as the immunogen. The resulting antibody distorted a planar porphyrin to facilitate the entry of a metal ion. 80 porphyrin molecules were metallated per hour antibody molecule, a rate only tenfold less than that of ferrochelatase. The uncatalyzed reaction was 2,500 times as slow as the antibody-catalyzed reaction. Antibodies catalyzing the kinds of chemical reactions exemplified by ester and amide hydrolysis, amide bond formation, transesterification, photoinduced cleavage, photoinduced dimerization, decarboxylation and oxidization have been produced using similar strategies. The power of transition state analog is now evident since they provide evidence insight into catalytic mechanism. They can serve as potent and specific inhibitors of enzymes. They can be used as immunogens to generate a wide range of noble catalysts. 3. Ribozyme. Thomas Cech 1983 found that the precursor of a ribosomal RNA in tetrahymena, a ciliated protozoan, undergoes self-splicing. The intron in this precursor RNA molecules is precisely removed by catalytic activity of RNA itself. This liberated intron then loses a short 5' terminal sequence to form a 385 nucleotide RNA molecule that catalyzes the transformation of other RNA molecules. This intron-derived RNA catalyzes the cleavage and joining of RNA chains at specific site without itself being consumed. Hence, it is true enzyme ribozyme. Another enzyme that contains a key RNA component is ribonuclease P, RNase P, which catalyzes the maturation of tRNA by removing nucleotides from the 5' end of the precursor molecule. Tetrahymena, a ciliated protozoan, a 414 nucleotide intron is removed from a 6.4 kilobits precursor to yield a mature 26 srRNA molecule. In a elegant series of studies of this splicing reaction, Thomas Cech and his co-workers established that the RNA spliced itself to precisely excise the 414 nucleotide intron. The self-splicing reaction requires an added guanosine nucleotide as an attacking group that becomes transiently incorporated into the RNA. G binds to the RNA and then attacks the 5' splice site to form a phosphodiester bond with the 5' end of the intron. This transesterification reaction generates the 3' O group at the end of the upstream exon. This newly attached 3. O group then attacks the 3. Splice. This second transesterification reaction joins the 2 exons and leads to the release of the 414 nucleotide intron. A phosphodiester bond is formed between the 2 exons, and the intron is released as a linear molecule. Like catalysis by protein enzymes, self-catalysis of bond formation and breakage in this rRNA precursor is highly specific. RNA precursors in the mitochondria of yeast and fungi also undergo self-splicing, as do some RNA precursors in the chloroplasts of unicellular organisms such as Chlamydomonas. Self-splicing reactions can be classified according to the nature of the unit that attacks the upstream splice site. Group 1 self-splicing is mediated by a guanosine cofactor, as in tetrahymena. The attacking moiety in group 2 splicing is the 2 O group of a specific adenylate of the intron. G1 conformation of proteins and polypeptides. Secondary structure. In 1951, Linus Pauling and Robert Corey proposed 2 periodic structures called the alpha helix, and the beta-pleated sheet. Subsequently, other structures such as the reverse turn and omega loop were identified. The alpha helix is a rod-like structure. A tightly coiled backbone forms the inner part of the rod and the side chains extend outward in a helical array. Alpha helix is stabilized by hydrogen bonds between the NH and CO groups of the main chain. In particular, the CO group of each amino acid forms a hydrogen bond with the NH group of the amino acid that is situated 4 residues ahead in the sequence. Thus, except for amino acids near the ends of an alpha helix, all the main chain CO and NH groups are hydrogen bonded. Each residue is related to the next one by a rise of 1.5a along the helix axis and a rotation of 100 degrees, which gives 3.6 amino acid residues per turn of helix. Thus, amino acids spaced 3 and 4 apart in the sequence are spatially quite close to one another in an alpha helix. In contrast, amino acids 2 apart in the sequence are situated on opposite sides of the helix and so are unlikely to make contact. The alpha helix, which is equal to the product of the translation 1.5a and the number of residues per turn 3.6 is 5.4a. The screw sense of a helix can be right-handed clockwise or left-handed counterclockwise. Essentially all alpha helices found in proteins are right-handed. In schematic diagrams of proteins, alpha helices are depicted as twisted ribbons or rods. The alpha helical content of proteins ranges widely, from nearly none to almost 100%. For example, about 75% of the residues in ferritin, a protein that helps store iron, are in alpha helices. Single alpha helices are usually less than 45A long. 2 or more alpha helices can entwine to form a very stable structure, which can have a length of 1000A, 100 nanometers, or 0.1 micrometers or more. Such alpha helical coils are found in myosin and tropomyosin in muscle, in fibrin in blood clots, and in keratin in hair. The helical cables in these proteins serve a mechanical role in forming stiff bundles of fibers, as in porcupine quills. The cytoskeleton internal scaffolding of cells is rich in so-called intermediate filaments, which also are 2-stranded alpha helical coils. Many proteins that span biological membranes also contain alpha helix. Beta sheets. Pauling and Corey discovered another periodic structural motif, which they named the beta-pleated sheet. The beta-pleated sheet, or, more simply, the beta sheet differs markedly from the rod-like beta helix. A peptide chain, called a beta strand, in a beta sheet is almost fully extended rather than being tightly coiled as in the beta helix. A range of extended structures are sterically allowed. The distance between adjacent amino acids along a beta strand is approximately 3.5a. The side chains of adjacent amino acids point in opposite directions. A beta sheet is formed by linking 2 or more beta strands by hydrogen bonds. Adjacent chains in a beta sheet can run in opposite directions, anti-parallel beta sheet, or in the same direction, parallel beta sheet. In the arrangement, the NH group and the CO group of each amino acid are respectively hydrogen bonded to the CO group and the NH group of a partner on the adjacent chain. In the parallel arrangement, the hydrogen bonding scheme is slightly more complicated. For each NH group is hydrogen bonded to the CO group of one amino acid on the adjacent strand, whereas the CO group is hydrogen amino acid. The NH group is hydrogen bonded to the CO group of one amino acid on the adjacent strand, whereas the CO group is hydrogen bonded to the NH group on the amino acid 2 residues farther along the chain. Many strands, typically 4 or 5 but as many as 10 or more, can come together in beta sheets. Such beta sheets can be purely antiparallel, purely parallel, or mixed. The beta sheet is an important structural element in many proteins. For example, fatty acid binding proteins, important for lipid metabolism, are built almost entirely from beta sheets. B. Tertiary structure. Myoglobin, the oxygen carrier in muscle, is a single polypeptide chain of 153 amino acids. The ability of myoglobin to bind oxygen depends on the presence of heme, a non-polypeptide prosthetic helper group consisting of protoporphyrin X and a central iron atom. Myoglobin is an extremely compact molecule. All dimensions are 45 times 35 times 25A, an order of magnitude less than if it were fully stretched out. About 70% of the main chain is folded into 8 alpha helices, and much of the rest of the chain forms turns and loops between helices. The folding of the main chain of myoglobin, like that of most other proteins, is complex and devoid of symmetry. The overall course of the polypeptide chain of a protein is referred to as its tertiary structure. A unifying principle emerges from the distribution of side chains. The second fact is that the interior consists almost entirely of nonpolar residues such as leucine, valine, methionine, and phenylalanine. Charged residues such as aspartate, glutamate, lysine, and arginine are absent from the inside of myoglobin. The only polar residues inside are 2 histidine residues, which play critical roles in binding iron and oxygen. The outside of myoglobin, on the other hand, consists of both polar and nonpolar residues. The space-filling model shows that there is very little empty space inside. The system is more thermodynamically stable when hydrophobic groups are clustered rather than extended into the aqueous surroundings. The polypeptide chain therefore folds so that its hydrophobic side chains are buried and its polar, charged chains are on the surface. Many alpha helices and beta strands are amphipathic, that is, the alpha helix or beta strand has a hydrophobic face, which points into the protein interior, and a more polar face, which points into solution. The fate of the main chain accompanying the hydrophobic side chains is important, too. An unpaired peptide NH or CO group markedly prefers water to a nonpolar milieu. The secret of burying a segment of main chain in a hydrophobic environment is pairing all the NH and CO groups by hydrogen bonding. This pairing is neatly accomplished in an alpha helix or beta sheet. Van der Waals interactions between tightly packed hydrocarbon side chains also contribute to the stability of proteins. We can now understand why the set of 20 amino acids contains several that differ subtly in size and shape. They provide a palette from which to choose to fill the interior of a protein neatly and thereby maximize van der Waals interactions, which require intimate contact. C. Quaternary structure. Quaternary structure refers to the spatial arrangement of subunits and the nature of their interactions. The simplest sort of quaternary structure is a dimer, consisting of two identical subunits. This organization is present in the DNA-binding protein CRO found in a bacterial virus called lambdaphage. More complicated quaternary structures also are common. More than one type of subunit can be present, often in variable numbers. Human hemoglobin, the oxygen-carrying protein in blood, consists of two subunits of one type, designated alpha, and two subunits of another type, designated beta. Thus, the hemoglobin molecule exists as an alpha-2 beta-2 tetramer. Subtle changes in the arrangement of subunits within the hemoglobin molecule allow it to carry oxygen from the lungs to tissues with great efficiency. D-domain structure. Polypeptide chains fold into two or more compact regions that may be connected by a flexible segment of polypeptide chain, rather like pearls on a string. The compact globular units, called domains, range in size from about 30 to 400 amino acid residues. For example, extracellular part of CD4, the cell surface protein on certain cells of the immune system to which a human immunodeficiency virus, HIV, attaches itself, comprises 4 similar domains of approximately 100 amino acids each. Often, proteins are found to have domains in common even if their overall tertiary structures are different. E. Reverse turns. Most proteins have compact, globular shapes, requiring reversals in the direction of their polypeptide chains. Many of these reversals are accomplished by a common structural element called the reverse turn, also known as the beta turn or hairpin bend. In many reverse turns, the CO group of residue N of a polypeptide is hydrogen bonded to the NH group of residue N+3. This interaction stabilizes abrupt changes in direction of the polypeptide chain. In other cases, more elaborate structures are responsible for chain reversals. These structures are called loops or sometimes omega loops to suggest their overall shape. Unlike alpha helices and beta strands, loops do not have regular, periodic structures. Nonetheless, loop structures are often rigid and well-defined. Turns and loops invariably lie on the surfaces of proteins and thus often participate in interactions between proteins and other molecules. The distribution of alpha helices, beta strands, and turns along a protein chain is often referred to as its secondary structure. G2. Ramachandran plot. The formation of alpha helix and beta strands is at the heart of folding process. We therefore need to understand the factor that influences the secondary structure. Let us begin by considering the range of backbone conformations that are accessible to protein. Examination of the geometry of the protein backbone reveals that the peptide bond is essentially planar. Thus, pair of amino acids linked by a peptide bond, 6 atoms lie in the same plane, the alpha carbon atom and CO group from the first amino acid and the NH group and alpha carbon atom from the second amino acid. The nature of the chemical bonding within a peptide explains this geometric preference. The peptide bond has considerable double bond character, which prevents rotation about this bond. The inability of the bond to rotate constrains the conformation of the peptide backbone and accounts for the bond's planarity. This double bond character is also expressed in the length of the bond between the CO and NH groups. The CN distance in a peptide bond is typically 1.32 A, which is between the values expected for ACN single bond 1.49 A and AC. The double bond 1.27 A. Finally, the peptide bond is uncharged, allowing polymers of amino acids linked by peptide bonds to form tightly packed globular structures. Two configurations are possible for a planar peptide bond. In the trans configuration, the two alpha carbon atoms are on opposite sides of the peptide bond. In the cis configuration, these groups are on the same side of the peptide bond. Almost all peptide bonds in proteins are trans. This preference for trans over cis can be explained by the fact that steric clashes between groups attached to the alpha carbon atoms hinder formation of the cis form but do not occur in the trans configuration. By far the most common cis peptide bonds are X-pro linkages. Such bonds show preference for the trans configuration because the nitrogen of proline is bonded to two tetrahedrocarbon atoms, limiting the steric differences between the trans and cis forms. In contrast with the peptide bond, the bonds between the amino group and the alpha carbon atom and between the alpha carbon atom and the carbonyl group are pure single bonds. The two adjacent rigid peptide units may rotate about these bonds, taking on various orientations. This freedom of rotation about two bonds of each amino acid allows proteins to fold in many different ways. The rotations about these bonds can be specified by dihedral angles. The angle of rotation about the bond between the nitrogen and the alpha carbon atoms is called phi. The angle of rotation about the bond between the alpha carbon and the carbonyl carbon atoms is called psi. A clockwise rotation about either bond as viewed from the front of the back group corresponds to a positive value. The phi and psi angles determine the path of the polypeptide chain. Are all combinations of phi and psi possible? G. N. Ramachandran recognized that many combinations are forbidden because of steric collisions between atoms. The allowed values can be visualized on a two-dimensional plot called the Ramachandran diagram. 3/4 of the possible phi-psi combinations are excluded simply by local steric clashes. Steric exclusion, the fact that two atoms cannot be in the same place at the same time, can be a powerful organizing principle. The ability of biological polymers such as proteins to fold into well-defined structures is remarkable thermodynamically. Consider equilibrium between an unfolded polymer that exists as a random coil that is, as a mixture of many possible conformations in the folded form that adopts a unique conformation. The favorable entropy associated with the large number of conformations in the unfolded form opposes folding and must be overcome by interactions favoring the folded form. Thus, highly flexible polymers with a large number of possible conformations do not fold into unique structures. The rigidity of the peptide unit and the restricted set of allowed phi and psi angles limits the number of structures accessible to the unfolded form sufficiently to allow protein folding to occur. Repeating values of phi and psi along the chain result in regular structure. For example, repeating values of phi -57O and psi -47O give a right-handed helical fold, the alpha helix. The cytochrome C256 shows many segments of helix and the Ramachandran plot shows a tight grouping of phi, psi angles near to -50, -50. Similarly, values in the region of phi equals -110 to 140 and psi equals +110 to +135 give extended chains with conformations that allow interactions between closely folded parallel segments beta sheet structures. The structure of plastocyanin is composed mostly of beta sheets and the Ramachandran plot shows a broad range of values in the -110 to +130 region. H1. Structure of DNA and DNA polymorphism. Utilizing X-ray diffraction data, obtained from crystals of DNA, James Watson and Francis Crick proposed a model for the structure of DNA. Subsequently verified by additional data, predicted that DNA would exist as a helix of two complementary antiparallel strands, wound around each other in a rightward direction and stabilized by H-bonding between bases and adjacent strands. In the Watson-Crick model, the bases are in the interior of the helix aligned at a nearly 90-degree angle relative to the axis of the helix. Purine bases form hydrogen bonds with pyrimidines, in the crucial phenomenon of base pairing. Experimental determination has shown that, in any given molecule of DNA, the concentration of adenine A is equal to thymine T and the concentration of cytidine C is equal to guanine G. This means that A will only base pair with T, and C with G according to this pattern, known as Watson-Crick base pairing. The base pairs composed of G and C contain 3 H bonds, whereas those of A and T contain 2 H bonds. This makes GC base pairs more stable than AT base pairs. The antiparallel nature of the helix stems from the orientation of the individual strands. On its position in the helix, one strand is oriented in the 5 feet, greater than 3, direction and the other in the 3 feet, greater than 5, direction. On its exterior surface, the double helix of DNA contains 2 deep grooves between the ribose phosphate chains. These 2 grooves are of unequal size and termed the major and minor grooves. This in their size is due to the asymmetry of the deoxyribose rings and the structurally distinct nature of the upper surface of a base pair relative to the bottom surface. Thermal properties of DNA. As cells divide, it is a necessity that the DNA be copied, replicated, in such a way that each daughter cell acquires the same amount of genetic material. In order for this process to proceed, the 2 strands of the helix must first be separated, in a process termed denaturation. This process can also be carried out in vitro. If a solution of DNA is subjected to high temperature, the H-bonds between bases become unstable and the strands of the helix separate in a process of thermal denaturation. The base composition of DNA varies widely from molecule to molecule and even within different regions of the same molecule. Regions of the duplex that have predominantly AT base pairs will be less thermally stable than those rich in GC base pairs. In the process of thermal denaturation, a point is reached at which 50% of the DNA molecule exists as single strands. This point is the melting temperature, Tm, and is characteristic of the base composition of that DNA molecule. The Tm depends upon several factors in addition to the base composition. These include the chemical nature of the solvent and the identities and concentrations of ions in the solution. When melted DNA is cooled, the complementary strands will again reform the correct base pairs, in a process is termed annealing or hybridization. The rate of annealing is dependent upon the nucleotide sequence of the 2 strands of DNA. Structural polymorphism of DNA. In addition to the major B form, 3 additional DNA structures have been described. 2 of these are compared to bDNA in figure. In variable humidity, the crystallographic structure of bDNA changes to the A form. RNA-DNA and RNA-RNA helices exist in this form in cells and in vitro. Short DNA molecules composed of alternating purine-pyrimidine nucleotides, especially Gs and Cs, adopt an alternative left-handed configuration instead of the normal right-handed helix. This structure is called zDNA because the bases seem to zigzag when viewed from the side. Some evidence suggests that zDNA may occur in cells, although its function is unknown. Finally, a triple-stranded DNA structure is formed when synthetic polymers of poly-A and polydeoxy-U are mixed in the test tube. In addition, homopolymeric stretches of DNA composed of C and T residues in one strand and G residues in the other can form a triple-stranded structure by binding matching lengths of synthetic poly-C-T. Such structures probably do not occur naturally in cells but may prove useful as therapeutic agents. By far the most important modifications in the structure of standard B-form DNA come about as a result of protein binding to specific DNA sequences. Although the multitude of hydrogen and hydrophobic bonds between the bases provide stability to DNA, the double helix is flexible about its long axis. Unlike the alpha helix in proteins, there are no hydrogen bonds parallel to the axis of the DNA helix. This property allows DNA to bend when complexed with a denebonding protein. Bending of DNA is critical to the dense packing of DNA in chromatin, the protein DNA complex in which nuclear DNA occurs in eukaryotic cells. Parameters of major DNA helices. Parameters in direction of helical rotation right-left residues per turn of helix 1110 base pairs rotation of helix per residue in degrees 33360 base tilt relative to helix axis in degrees 2067 major groove narrow and deep wide and deep flat minor groove wide and shallow narrow and deep orientation of end glycosidic bond anti for py sin for poo. Comments. Most prevalent within cells occurs in stretches of alternating purine-pyrimidine base pairs. H3. Structural polymorphism of RNA and 3D structure of tRNA. Three kinds of RNA molecules perform different but cooperative functions in protein synthesis. They are mRNA, rRNA and tRNA. 1.